Assalamu alaikum everyone, this is Nasir Ali here. Dear student, how are you? Hope you will find by the grace of Almighty Allah. Dear student, our today topic of discussion is antifungal drugs. But before that, I am going in the detail of the antifungal drug. You people should know about the fungus. That's what's mean by a fungus. So fungus is a eukaryotic organism. So now what's mean by a eukaryotic organism? Those organisms which have a well-developed nucleus and a cytoplasmic organelle is known as eukaryotic organism. So fungus is a eukaryotic organism and having diploid number of chromosome and cell wall, and fungus have a diploid number of chromosome, full number or actual number of chromosome, not the diploid number of chromosome, and having a cell wall made of protein and chitin. Fungus is a having diploid number of chromosome and a cell wall which made up chitin and protein, and may consist of a single or multiple cell, or may exist as a single cell or multiple cell. And if the fungus is exist or consist of only a single cell, then fungus is known as yeast, like Candida species, Pneumocystis species, and Cryptococcus neoformans species. So when the fungus exists as a single cell, then the fungus is known as yeast, and the example of such a fungus is Candida, Pneumocystis, and Cryptococcus neoformans, and such a type of fungus exists as a single cell and known as yeast. While if the fungus consists of a multiple cell, then known as modes like Aspergillus, Rhizopus, Mucor, and Absida. So such a type of fungus is known as mold. And example of such a type of fungus is Aspergillus, Rhizopus, Mucor, and Absida. So that's all about the general introduction of the fungus. That fungus is a eukaryotic organism having a diploid number of chromosome and cell wall, and may consist of a single cell or multiple cell. If fungus consist of or exists in a single cell, then known as yeast, like Candida species, Pneumocystis species, and Cryptococcus neoformans. While if the fungus exists or consists of a multiple cell, then known as molds like Aspergillus, Rhizopus, Mucor, and etc. So that's all about the general introduction of the antifungal drug. Now move forward toward the structure of the fungus with respect to pharmacological target. In today's lecture, we will discuss the structure of the fungus, but with respect to its pharmacological target. So pharmacologically, the fungus have three main parts. Pharmacologically, the fungus have three main parts: cell membrane, cell wall, and nucleus. And all antifungal drugs are classified according to the different structure of the fungus on which they are act. So pharmacologically, the fungus having three main parts. The first one is cell membrane, the second one is cell wall, and the third one is nucleus. So we will discuss each part in the detail. The first part is a nucleus. The first part is a cell membrane, and the main component of this cell membrane. This is the cell membrane of the fungus. This is the cell membrane of the fungus, and the main constituent, the main part of this fungus cell membrane is the ergosterol, and it is a unique component of the fungus cell which distinguish the fungal cell membrane from the human cell membrane. And this ergosterol is made of the squalene. Squalene is a substance which is present in the fungus cell, and this squalene is converted to the squalene epoxide by an enzyme known as squalene epoxidase. And this enzyme converted the squalene into squalene epoxide. Then the squalene epoxide is converted into the linosterol, and linosterol is converted into the ergosterol by an enzyme known as protein alpha demethylase methylase demethylase enzyme, which is responsible for the demethylation of the linesterol. This linesterol is converted by protein alpha demethylase into the ergosterol. And how this linesterol is converted into the ergosterol, this is actually done by the demethylation of the linesterol into the ergosterol. And now this ergosterol provides structural integrity into the cell membrane. So this is the first part of the fungus cell membrane. The second part of the fungus cell is cell wall. This is the cell wall of the fungus cell, which made of different layer of protein and chitin. And these layers, the protein and chitin layers, are linked together by the beta glucan. This is the protein layer and this is the chitin layer, and this link is a beta glucan, which is responsible for the connection, which is responsible for the linkage of the protein and chitin layer. And this beta glucan, these small parts are the small transition compounds, which is known as beta glucan, are synthesized by the beta glucan synthase. This is the beta glucan synthase enzyme, and this is beta glucan, and this beta glucan then link the protein and chitin layer with each other and provide structural integrity into the fungus cell. So this is the second part of the fungus cell, which is cell wall. The third part of the fungus cell is nucleus. This is the nucleus of the fungus cell. And the nucleus of fungus cell, we will actually discuss two classes of the antifungal drug. The first one is those drugs which are bound to the microtubule. The first one is in the nucleus of the fungus. We will discuss about the microtubules, and the second classes of the drugs is drugs which is anti-metabolite drugs anti-metabolism. And the nucleus of the fungus cell, we will actually discuss about the drugs which are attached, which are bound to the microtubule, and drugs that are responsible for the anti-metabolite mechanism or procedure, which is responsible for the reversing the metabolism of the, or which is responsible for the inhibition of the metabolism of the fungus. So in the structure of the fungus with respect to pharmacological target, we will actually discuss three main parts: cell membrane. And cell membrane, the main constituent is the ergosterol. You people should remember the mechanism of the ergosterol synthesis, that ergosterol is formed from the squalene. Squalene is converted to squalene epoxidase, squalene epoxide by the enzyme known as squalene epoxidase, and this squalene epoxide is converted into the linesterol, and the linesterol is then converted to the ergosterol by the enzyme known as protein alpha demethylase enzyme, and then this ergosterol is embedded in the cell membrane and provides structural integrity into the cell. The second part is the cell wall, which made up the protein and chitin layers, and these layers are are connected to each other by the beta glucan synthesized by the beta glucan synthase enzyme. And the third part is the nucleus, and nucleus we people actually discuss about the microtubule and those drugs which bind to the microtubule and anti-metabolite drugs. So that's all about the general introduction of the fungus and structure of the fungus with respect to its pharmacological target. So I hope you will understand. Otherwise, if you people have any questions regarding today's lecture, so you people should ask me in the comment section, and in the next lecture we will discuss about the antifungal drugs and classification of the antifungal drugs. So thank you so much for giving me your useful time. Thanks.